A very good afternoon. It's my pleasure to introduce, on behalf of Rajiv Varshni, Dr. Elizabeth Jones. Liz has been currently appointed as manager for the GOBII project at Cornell University, USA. And to introduce her, I'll give a brief background about her past positions at Pioneer and Syngenta. She is the first to implement high throughput seed genotyping laboratory and develop marker protocols to characterize and manage seed purity and protect germplasm at Pioneer Seeds. And at Syngenta, she has been involved in development of marker systems that can meet breeder needs for high quality data in short time frame. In both these industry positions, the development of efficient database systems have been the key for her role at current position at GOBII project in Cornell University. And I think Liz is not new to ICRISAT because she has been here as a student for PhD. She has been working in collaboration with Centre for Arid Zone Studies, University of Wales and Cambridge Laboratory, and in the Pearl Millet Group at ICRISAT, India. So I would like now to invite Liz for her presentation. I think you have changed your title, so
I forgot what I gave you for a title.
Okay. Okay. Please join me in welcoming Liz for the today's presentation.
Thank you, everyone. It's an honor to be here today. Today I'm going to be talking about how markers have been used and how they've changed their use in molecular breeding and how the perspective of using markers has changed. Can you hear me at the back? Yep? Okay. And I wanted to start out with my PhD research. It's going back a long way now, but it's an old story, but it's a good story. I started out working at ICRISAT, and it's probably the reason that I'm back here now, because I had such a good time during my research that I wanted to start working with the CG centers again. And when this GOBII position came up, I jumped at the chance to be able to leave industry and start working with the CG centers again. So pearl millet, as many of you no doubt know, is a staple food crop of the semi-arid tropics. When the first hybrids were developed, it succumbed to downy mildew disease, causing massive yield losses. And at the time, very little was known about the genetics. And also at the time, markers were just being used for QTL mapping, and this was thought to be the answer to understanding the genetics of this system. It's a quantitative trait, and it was known to be regionally variable. So we started out by collecting pathogen populations from Africa and from India, and screening them in two locations, well, several locations. The first one was in Wales, and that's me as a PhD student counting pearl millet plants in Wales. And the reason why it was Wales, which is possibly the rainiest place in the world, it's like Seattle for the level of rainfall that there is, was because you could spray the pathogen there and have no danger of infecting any crop. And then in ICRISAT, we were screening just against the local pathogen populations. And this is basically the culmination of the research. Here I'm showing the linkage groups, which at that time were mapped with RFLPs. And the threshold for LUD score is 2 or 2.5, and we were seeing these huge QTL peaks. And these are the QTLs for resistance to pathogen populations in India, Niger, Nigeria, and Senegal. And you can see very distinct clear peaks. Each one has very distinct avirulences and very distinct resistances in the pearl millet lines. So this is the first evidence that really there is a race or host pathogen specific system happening in pearl millet. The QTLs were also extremely stable, very high heritability. You see different disease screens here. In the UK, it was very hard to get enough of the pathogen population growing to be able to get very high inoculum load. In India, it was very high, but still we had very consistent QTL. Now, as an aside, I was most fascinated by these little QTLs. I didn't know whether they were modifier genes or they were just avirulences in the pathogen population at a low frequency. So the last part of my PhD, I spent trying to isolate the individual, to basically isolate A. virulences from the whole pathogen population. And I don't know whether any of you have seen the pathogen under the microscope. They have zoospores, which swim very fast, and it's very hard to catch them. So I spent a lot of time trying to catch single zoospores. In the end, went with the dilution method, grew pelmet seedlings on agar and diluted the pathogen to infect them, and then looked at whether I was going to pick up different QTLs spraying with an individual isolate rather than the whole pathogen population. And here you see for the individual isolate from the Nigerian pathogen population, this is the whole pathogen population, and light blue is the effect from a single zoospore isolate in exactly the same QTLs, so I wasn't seeing anything different. I was expecting like one of these little peaks to suddenly become a big peak if I'd isolated A virulence with a very low frequency. And then when I looked at another isolate, I saw nothing at all. And for a while this was quite perplexing until I started to look at my data, and we had tall chlorotic phenotypes and then also these stunted phenotypes for susceptibility. And when I analyzed these two phenotypes separately, I got two major peaks in a completely different place to the original pathogen population. So I was isolating new A virulences. And the effect of those two scored together were cancelling each other out. So if I was scoring total downy mildew, my susceptible parent alleles were equivalent to my resistant parent alleles. If I scored these two separately, I was getting a difference between these two alleles. So that's just a nice genetic story. Started my career on a nice path. I was very confident the markers were going to be able to solve everything in breeding. And also led eventually to an improved variety of corn millet. Then I moved on to a postdoc, and this was a very different situation. Now I was dealing with a very environmentally variable trait, and this was resistant to Phytophthora infestans in tomato or late blight resistance. And it was well known that late blight resistance is very short-lived in potato, only lasts for 1 or 2 years. And so in tomato we were looking for some more durable resistance. Now this is tomato variety that we were using for breeding. It was a wild variety of tomato, a Phytophthora, that was resistant to over 100 isolates. And we classed this to a susceptible cultivated variety of L-Esculentum. And you can see that these have both been inoculated, and this one's suffering considerably. Now, unfortunately, as happens with many of these grants, we didn't have time to make a very suitable population, and so the quickest population that could be made was just a bidirectional backcross. So we were screening pathogens, and we had races of pathogens on a backcross to the hirsutum and a backcross to the L-Esculentum. And I don't have any pictures of this, but the hirsutum plant is a wild, crazy thing that just grows and grows and grows, whereas Esculentum is a determinate plant and very tidy. So the architecture of these two plants are very, very different, and screening them in this first rookie state, basically, we were screening them in backcross populations where the phenotype had a major effect on what we were trying to score, on the disease we were trying to score. And so we got QTLs. Everyone can get QTLs. If you throw a pathogen at some plants, you're going to get some QTLs. They're extremely environmentally variable. We've got 12 chromosomes in tomato, and pretty much every chromosome had multiple QTLs on it. And most of those QTLs were associated with some kind of canopy or plant architecture effect. And there was one very interesting QTL that I'd just like to talk about, and it was a QTL associated with droplet dispersal. So in the cultivated Esculentum, you put a droplet of inoculum on the leaf, and it stays there because of the surface tension of the leaf. Whereas in hirsutum, these have been inoculated with exactly the same inoculum droplet. The droplet disperses, and so it doesn't get the chance of the pathogen to infect the leaf, presumably, and that's why they're resistant. And this is quite possibly because of the differences in trichomes that you get in Esculentum versus hirsutum. Esculentum has mainly type 4 and type 6 trichomes, a mix of
And so you get very different effects of surface tension. And it would have been really nice to investigate this further and see whether this trichome density and differences in trichome could be actually implemented in breeding. But this was after I'd actually left the postdoc project at this point. So this is a series of neurosogenic lines. Someone put a huge amount of work into this, and they took the 4 major QTL, including that one associated with droplet dispersal, and they introduced them into an esculentum background in groups of 1 to 4, and screened those in multi-locational trials. Huge amount of work went into this, but in the end, nothing came out of it. So every one of those QTLs was associated with some negative effect on yield, on plant architecture, and was undesirable to be sold commercially. So I spent some time in Australia working on similar types of projects, but I was getting very quickly frustrated with working on QTL mapping, finding QTLs, but then nothing actually being implemented. After Ithaca, which was my first success story, things really went downhill, and it felt like I wanted to go into industry to find out how industry were implementing markers in breeding. So I worked 10 years at Pioneer and 5 years at Syngenta, and as you probably know, I can't talk about specifics here, and so this is more like a generalization of the work that I did there. So I joined Pioneer at a time when they were just switching over from SSRs to SNPs, so I set up the SNP lab there. This is the evolution of marker systems at Pioneer, went from epiparacles to SSRs to SNPs. Isozymes lasted a long time. In fact, I think they might still be going, and I'll tell you why in a little bit. And markers were being very effectively used with the SNP markers. We were using them for marker-specific selection, and we were using them mainly really for transgene integration. But major genes, major QTL effects, we weren't dealing with anything that was quantitative or environmentally variable. Now, another area that we were working on, and this was a huge project both at Pioneer when I was there and also at Syngenta, was developing marker sets for seed purity. And at Pioneer, I can talk about how we did this because we talked about this publicly. We used an algorithm called the traveling salesman problem, and this is an algorithm whereby you're trying to find the shortest route for a salesman to go through every point that he needs to go to deliver his products, to sell his products. And this is the TSP, the traveling salesman problem, applied to all the British Isles tourist attractions. So it's being used to show the shortest route to be able to go to every tourist attraction in the UK, and I think it's pretty interesting that they go to Ireland twice. I'm not sure they thought about the ferry routes here, but it can be applied to a number of different problems. And in fact, in the marker lab, we were using this, the TSP, to define the smallest number of lines that we could use in the lab QC to be able to distinguish all our markers, basically to check we had the right marker to the reagent to be able to run markers. And so then we turned it around and said, well, we can also use this to select the smallest number of SNP markers to distinguish between all of our lines, and that's what we did. This shows the number of markers that you would need to distinguish between lines, and this is the number of inbreds. At the time, we had about 400 standard inbreds that we had SNP data on. And you can see that so about 5 markers distinguishes about 20 or 30 inbreds, and when you get up to about 400 inbreds, you need about 15 markers. And in fact, you can see the slope levels out. So in fact, you could probably use about 12 markers to distinguish about 99% of the pairwise combinations of inbreds. So we had a very large seed purity lab at the time that ran isozymes, and isozymes were the standard globally for using in seed purity tests. And there are very large group of very competent people who were running these isozymes, and they were very reluctant to switch to SNPs. And especially because SNPs had, that dropout. You're going to get that missing data. And isozymes consistently give you results. So they said there's no way we can move to SNPs because of the missing data. So we did some tests on real life seed lots. We actually ended up selecting 16 SNPs just because it fits into the robotic format that works well in the lab, and compared them to the standard set of 15 isozymes that we used at the time. So this shows average number of matching index, and I'll explain this a little bit. So this is the isozyme profile, and this is the SNP profile. So with isozymes, if you have 15 isozymes, for one inbred, you'll get an average of about 45 other inbreds in the set that have exactly the same isozyme profile. Whereas SNPs, you get 0. So the SNPs uniquely distinguish amongst everything. And if you increase the amount of missing data, the SNPs are actually very stable. Don't increase the number of matching inbreds hardly at all, whereas the isozymes go up. And even with misscoring, so now we're looking at misscoring, so this is just artificially entering the wrong allele into the system. And you can see that again, SNPs is actually stable even with misscored SNPs. And it's only once you get to 4 or 5 misscored SNPs that you start to get other matching inbreds, so false matches of inbreds, whereas the isozymes are very susceptible to misscoring. So we showed that even with missing data and even with misscored data, that SNPs were extremely stable. And this is because there's a certain level of redundancy in 16 SNPs. You can have several drop out, and the rest of the markers basically can still distinguish amongst the lines. Another large project that we worked on that I can also talk about is using markers in plant variety protection. And we had a big project run through ASTA, which is the American Seed Trade Association, to look for a set of markers that could be used in intellectual property. And this was after the era of what was called flashlight breeding in the US. Pioneer has, I would say, maybe I'm biased, but the best germplasm in the world for maize. And there was a series of people going into fields at night with their flashlights and stealing plenty of lines. Now, you can use markers to show that someone has stolen one of your printer lines. We had several cases that proved that. But what if someone's taken your lines and then done one backcross and slightly moved away from it? And that was what was also happening in a lot of cases. So we used markers to find thresholds above which basically your if someone took your variety and it was too close to your variety, that it was considered an EDV. EDV concept is a little hard, the legal language is a little hard to get through, but an EDV, or an essentially derived variety, is a variety that is predominantly derived from the first variety, so basically it's bred from the first variety, but retains the expression of the essential characteristics that the first variety has plant variety protection. So if you have variety A, plant variety protection, and variety B, then you can prove whether it's too close to the first one using these essential characteristics. But legally, it's extremely difficult to say what the essential characteristics of a plant. And there's been several well-known cases for gypsophilia, which is a flower. It's that fluffy, I don't know if you get it here, the fluffy flower that is used as filler in bouquets of flowers. A case that's gone on for 15 years now, arguing about whether variety B of gypsophilia is essentially derived from variety A. So breeders and the whole industry wanted a clearer demarcation of what was the same or what was different for a variety. And after several years, I would say 10 years of working on this project, we finally came to an agreement on the marker set and also on the threshold that we could use to show whether something was an EDV. And it's a set of 3072 alumina markers that were selected in the end, and these were agreed on by not only all the companies in the US, but all the companies in Europe to abide by as well. And using this marker set, anything, so you did your genetic distance analysis and you had to use a specific genetic distance analysis. It was Rogers genetic distance analysis. Any pair of lines less than 91% distance were considered far enough apart. They're outside of the EDV zone, so they were okay. Anything greater than 91% is called the reversal of burden of proof. And this means that it's the company that's potentially stolen your line that has to prove they didn't steal it. So the burden is on them to prove they didn't do it. And then above 95% is a definite EDV in France, but in the US you can still go to court and prove otherwise. So this was the first set of markers that have ever been used to distinguish between lines and use in the EDV concept. Several other crops are following on from this. The next part of this project, and I would say this project is where I met Kelly Robbins, who actually now runs the GOBII project. He did the data analysis on this project. And we both left before the project moved on to actually using markers in plant variety protection. And I can't remember, there's an equivalent system in India. So this is where you register your varieties. It's not so much a seed registration, but a legal protection of your varieties. And millions are spent on collecting all the phenotypic characteristics that go into the application that goes to the government for plant variety protection. For maize, it's over 50 characteristics have to be collected. So this project looks at whether we can use a marker set instead of the phenotype to be able to register varieties legally. Okay, so back to breeding. So it was all going swimmingly for, as I said, single gene traits, seed purity, fantastic, EDV, fantastic. But for QTLs with environmentally variable effects, there were just too many. So Pioneer had a really nice database of QTLs, and it was like Christmas lights. Every chromosome had multiple QTLs on. Some had positive allelic effects, and some had negative right next to each other. So it was really overwhelming for breeders to actually handle this level of data. You know, I select for this QTL, but then I have a negative effect on disease. How do they even begin to manage that level of information? You know, we're very lucky that around that time, when everyone was getting overwhelmed with QTLs, that the concept of genomic selection came along, and it just completely changed the landscape. Instead of having to think about all of these different QTLs, genomic selection would basically just tell you which lines to cross that was going to improve your yield. So this is a complete game changer. Now I also want to mention that companies also went in a reverse direction too at this time to gene modeling. And I've not been involved with gene modeling. This is kind of the cool kids over in the other house that have a big trampoline, and you look at them very enviously. But watching what they're doing all the time. The gene modeling systems that are being put together now are very sophisticated. Basically, you take all of your data, all your QTL data, all of your biochemistry data, all of the public information out there, and you put a hypothesis together on what are the most likely sets of genes that are going to be key for any particular trait. And then you can use tilling to test your hypothesis. Tilling is where you do the mutation screening, or now you can do gene editing to test your hypothesis, or you can use eco-tilling to look for variants of those genes, so look for whether there's any natural variation for those genes. So back to genomic selection. Everyone was very excited at the time because not only did we have genomic selection, but we had sequencing costs were coming down. GBS was reducing the amount of the genome we had to sequence. And so everyone was saying, well, let's just sequence everything. You know, we've got the money, we'll sequence all of our breeding lines, and we can do these fantastic genomic selection programs. And this is a landscape of the marker systems that have been available recently. You have a number of samples on the x-axis, number of markers on the y-axis. So for large numbers of samples, small numbers of markers, you know, we have CASP or TACMAN or other types of marker systems. And then smaller number of samples because it's more expensive. We have high-density chips like Axiom, medium-density chips like Illumina, and we had sequencing for the super high-density markers, millions of markers. And really everyone thought, well, GBS is just going to take over all of this. We don't need chips anymore. We can just sequence everything. And this is my analysis of chips versus GBS a few years ago. Chips was really considered its old technology. It's not going to be around much longer. Instrumentation is expensive. It's going to get outdated very quickly. The high development costs, you have to sequence your lines first to find your SNPs to put on the chip. And then there's ascertainment bias, because due to the lines you've sequenced, you're going to get a bias in the polymorphisms on the chip. And it's much harder to capture haplotypes. Usually your SNPs are separated across the genome. With GBS, next generation technology, so always sounds good. It's the next generation technology. Costs are going to kind of keep coming down. The development costs are very similar. There's pretty similar protocols on different crops, different heterotic groups, different germplasm groups, and very limited ascertainment bias. There is some ascertainment bias due to the reference genome that you're using, but generally, other than that, very little. And you can capture local haplotypes. You've got your sequence, you can capture haplotypes, you can capture a lot of information. But in reality, the landscape did not prove in the next few years, it disproved my hypothesis. Chips, very savvy, especially Illumina, chip costs kept coming down and down and down, and were lower than the costs of GBS. They really recognized the agricultural market, and they really moved towards the agricultural market. For high volume of samples, they would reduce the price of the chip. They started making higher throughput platforms for the chips as well. With GBS, we still struggled with the library, making the libraries. DNA preps will still have to be higher quality than we were using for chips, and so that was expensive too. And then the cost of actually processing the data was very high. People kept saying, but what's the cost of capturing that amount of information? And we put a project together to assess it, and it was actually pretty high compared to an Illumina chip. The amount of storage and processing that you had to go into GBS data was pretty high. And then, of course, we had FTO issues. P-Gene had some patents in this area, and they started approaching companies and saying, look, you can't be doing this. And Cornell stopped running GBS for us. And in fact, recently, P-Gene's called sequencing by genotyping patent has been upheld in the US recently. So it's put a stop to any of the GBS applications. There are other technologies coming up, but P-Gene's been quite aggressive in this area. But really, the readers are saying, we don't want all of the sequence data. It's too much information. We don't think we need that much for genomic selection. And this is a paper from Zhao et al. And it basically shows your prediction accuracy, and this is in Maize germplasm, and the number of SNPs. So it levels out probably about 600 SNPs, but 300 to 400 SNPs. That's enough to give you a significant prediction of yield. So readers were saying to us, as marker lab, we would say, well, we can give you 10 times the SNPs for the same price. And they're just saying, we don't care. We want the same SNPs, and we want lower price. And at the time, the industry was going through quite a change. It was been through a boom time. And then in 2003, the market started to collapse, especially due to South America. There's a slowing growth worldwide. And there's a huge amount of pressures on breeders to reduce costs. It's exactly the same as been happening at the CG centers. So breeders were coming to us and saying, well, I really like running my plots, but I'm quite happy to give up running plots if your market prices are cheaper than the price of a plot. And it depends on the plot, but it can be about between $5 and $15 for a plot. Now, previously, the plot was still lower cost and had high G-value. But the markers were much higher cost than running a plot, even though there's no G-value. So they're a bit like, oh, well, we'll rather run the plots right now because I like to see my plants anyway. But with the pressures on the budgets, they started to say, well, we want you to be at the cost of growing a plot out, and then, not long after that, we want you to be a lot less than what it costs to grow a plot out. So we were looking at markers, technologies, and really there's a big gap. We look at this, technology that will fit that 300, 400, 500 mark, that you can run on millions of samples, hundreds of thousands of samples, and is very, very cheap. And an emerging technology a few years ago was, it has several names. This is next generation genotyping technology. It's also called targeted sequencing, and it's sometimes called GBS. And this is where it's like a traditional marker system. It's primers and probes. You target the regions that you're interested in. You can target your traits, but you multiplex them together, and you multiplex your samples, and you put them all in one tube, and you sequence them. And because you're not sequencing many regions, you can really plex very highly in the samples. Most of these amplicon or ligase-based technologies, or I have seen some other chemistries out there too. Affymetrix, PGenes, and Illumina all have technologies that they're starting to promote quite heavily in this space. And this just shows an example of amplicon sequencing. So you just design primers to each of your regions, and you pull everything together in a tube, and your samples, you add a barcode to, you pull everything together and sequence it. So it felt like we had the chemistry, pretty much the right chemistry in this space, and it was a good time to be developing this chemistry. We were getting a lot of requests for 2-week turnaround time, and that included shipping and DNA prep and data analysis. So basically, for the marker technologies, you had to be done in 2 or 3 days. You had to be through the lab processes in 2 or 3 days. And that was usually we were working a lot with double haploids, so it was in the space that you had to transplant the double haploids, you had to have those results. It's also the case for planting any kind of seedling. The cost of putting these things in the field or transplanting them is very high, so you have to have the markers back in that time. So really genotyping at that time was becoming much less about chemistry and biology, much more about logistics, more like just understanding shipping requirements, understanding phytosanitary requirements, being able to coordinate and batch samples together so that you could get the cheapest price for shipping, that you could get the cheapest price for automation and running your SNP markers. A big part of this is data management, and so this has become a huge focus of any genotyping lab now, is the data management. And not just having a data management system for the markers, but be able to have end-to-end data management from your germplasm to your samples, taking your samples, running them in your lab, and making decisions about which ones are going to be selected. So a total decision support system. And there are also lots of decisions that have to be made about where you're going to put your central hubs, where you're going to have your local hubs, and how you're going to connect all of these systems together. Now, I won't dismiss GBS or sequencing totally. GBS has a big role to play in discovery still, in discovering new SNPs or germplasms that have been very under-characterized. You can go in GBS and really understand a lot about new germplasm groups. And also there's new computational methods that are very exciting right now. I don't know whether you've heard of Energene, but those kinds of companies that are developing algorithms to turn a sequence into haplotypes. And haplotypes is a much easier form of information to understand than SNPs. And you can start, instead of looking at SNPs and inheritance of SNPs, you're looking at, with IVD or identity by descent, you're looking at recombination segments in your breeding population. And this is, I think, a very exciting area where sequencing can be used. I would say that for a lot of the applications that breeders have, you don't have to be very complicated in the IVD algorithms you're using. You usually just have to go back to grandparents and understand the inheritance and the combination as well, you don't need to be working with physical maps necessarily. You don't have to align everything to a physical map. If you have a population, you can look at the co-segregation of haplotypes and create a genetic mapping of the population you're working with. And then it's not biased by the reference genome you're using. Okay, so this is my last slide, and I really see that molecular breeding is going to become simplified again. We've been through, I think, 5-10 years of just too much information, too much QTLs, too much sequence data, too many SNPs, and I really see that with the new marker systems, it's going to be simplified information, simplified decision support for breeders. There's a lot of work to do on the lab side in terms of data management pipelines and decision support systems, and that's why I'm in the GOBII project now, is to build those kinds of systems. There's definitely improvements in sampling needed. Surprisingly, that's the big bottleneck now. We have most companies, most people have developed very cheap, very good DNA preps. That used to be the bottleneck. Now with those in place, now we're moving back to sampling. How do we sample hundreds of thousands of plants efficiently and get them to the lab efficiently? Now I think we're going to see continuous improvement still in the genetic space just through those modeling systems I was talking about, and of course gene editing is a big space that we're going to see exciting things happening in the future. And that's it. So thank you very much.
Now the floor is open for questions. Manish.
Okay, nice presentation. Actually, I have two questions. One is related to EDVs. So, in EDVs, like you mentioned that a particular company has come up with a set of SNPs, and whatever lines they have, they know how much percentage genome based on those SNPs are available. And if someone other is doing, say, cosmetic changes, 1 or 2 traits and all those things, so that can be caught. So that panel is for company by company, or it's for crop, as per crop?
No, it was a single panel that was decided upon across all of the companies. So you have to use the same SNP panel for every variety, whatever company you are coming from, in Europe or the US.
Okay. Okay, so other question is on genotyping platform. So just I want to know, like, especially for genomic selection. So GBS has entered in genomic selection first, and in trait mapping also it is doing very good. But what actually the disadvantage is lots of missing data coming up. So even if you take a biparental population or any panel, due to lot of missing actually SNPs which you are using in trait mapping, that is getting reduced drastically.
Right.
So that is like shortcoming coming in trait mapping, and if we apply, say, in genomic selection as per se for array, so at least their number is fixed, but they are giving, like, say, consistent data for a particular loci. So when you analyze your data, you know what you are going to infer from those data, whether it's a trait mapping or breeding. So just I'm little bit biased more towards arrays than the GBS. So I want to just hear from you.
Yeah, that's certainly an advantage of using a fixed marker set, is the consistency of data that you get back. And it was certainly, trying to switch to GBS, that was the major pushback we had from breeders, is they wanted the same marker set that they've already been using because they had all the historical data on that same marker set. So, we put quite a big effort into converting the GBS data into the standard marker data, but that's all time and it's processing power and it's cost in the end. It's certainly being used and it's being published. To what extent, I couldn't really say. I mean, it's certainly being used. Different methods are being And it has different successes in different crops. I mean, Syngenta is a huge company, well, not a huge company, but had 20 different crops. Maize, you know, it works great. Other crops really, it's a struggle. So it's definitely a crop by crop thing.
Hi, Liz. My question is related to Manish. Another Manish questions. First, that you told that markers for the PV, plant variety production. So is that got a legal value in the US if you have the markup data for the plant variety production?
So that's a project that's just started, and that will probably be another 10-year project before they come up with legally what marker set.
So still it's not legal.
No, for
Legally,
EV it is, for showing whether a line is the same or different to another line. To actually protect your plant, that's another, it's a government system. It takes a long time to make changes, so it'll be a 10-year project before that's solved.
My second question related to genomic selection. You have suggested the number of markers, 300 point markers. What are the number of lines in the training population? How many lines you feel that makes a good training population?
It depends on the diversity of the material that you're working with and how closely related the populations are. So I can say several hundred to 1,000.
Thank you.
Mm-hmm. What's the algorithms? So the algorithms are proprietary, so I don't think any of those are published. But basically you just put all the information you can find into a big pot and then run these algorithms and then come up with the best hypothesis of what genes are most important. And it seems to be pretty successful.
Thank you, Liz, once again for your nice presentation.